Hmm. Hmm?
Ma'am, I am presenting the screen.
Okay, just wait. Can you see the screen? Can you all see the screen?
Yes, ma'am.
Hello. Yeah.
Yes, ma'am.
So
Display
you
is
have
visible.
enzyme plus substrate and inhibitor. Okay. When you have substrate plus inhibitor, if substrate bind to enzyme, what do you get? If substrate bind to enzyme, what do you get? What is the ultimate reason for binding of substrate to enzyme?
Product.
Very good. You get product. If enzyme binds to, if inhibitor binds to enzyme, what do you get? Do you get product, Shifa? If inhibitors bind to enzyme, do you get product? Can you all hear me? Shusti, can you hear me? Yes. If enzyme binds to inhibitor, do you get product? No. There's no. What is inhibitors? Listen, inhibitors a substance which prevents substrate to bind and convert into product with the enzyme. So if substrate binds to enzyme, you get product. If substrate doesn't bind, in the presence of inhibitors, it is said to be it's no product. Okay, those are inhibitors. So the different theories, one is competitive inhibitors, other is non-competitive inhibitors. So first we'll be studying the competitive inhibitors. See, competition is between, in the class, a student, A student and B student, and there is C student. This student score around 95, this around 99, and this is somewhere in the 60. So the competition is between whom? Whether it is in competition is between AB or AC or BC or BA. A and B, right? The competition is between A and B. Same way, here what happened? Substrate and inhibitors are almost similar structure. They're almost similar structure. When the substrates and inhibitors is almost similar structure, there is a competition whether the inhibitor binds or substrate bind. So they compete to binding on the active site on the enzyme. Clear? Now, this reaction can be reversible. What do you understand by reversible? If you have more of substrate in your reaction, then it will not allow the inhibitors to go and bind. So if you have more of substrate, then substrate only goes and bind to the active site. So this reaction can be reversible if you add more substrate. If you, on increasing more substrate, increasing more substrate, you can reverse the reaction on increasing more substrate. Now, if you remember the graph, last time we have drew, this is one, this is one, this is, and this is a substrate, This is a velocity. This is the maximum velocity, that is Vmax. This is a km, 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 km dash, because it's a new km. This one is without inhibitor, without inhibitor, and this line is with inhibitor, right? So the km for both. So in this reaction, does Vmax change in the presence or absence of inhibitor? Does Vmax change? So Vmax remains same. Does km change? Does the km change? Does the km change? Yes. Km changes. There's a difference. Km and km dash. The km changes. So this is called a very good. Vmax do not change and km changes. This is a competitive inhibitors. Clear to all? So km value increases. In presence of inhibitor, km value increases. Write down. Now we'll be studying few examples which are important that you should know. Please write down this and let me know. Then I can start. Are you all done? Or do you need some time? Are you all there? Hello. Can you all hear me? Hello.
Yes, ma'am.
Kanika. Yeah. Are you able to hear me?
Yes, ma'am.
Okay, fine. I think I'm getting the messages lately. Okay. Yeah. So next. Next. Can you see the screen? Can you all see the screen?
No, ma'am.
Okay, now can you see? Now can you see?
Yes, ma'am.
Okay, write down the examples. These are the examples for competitive inhibition. Here, succinate is a substrate, fumarate is a product, and then you have succinate dehydrogenase. Succinate dehydrogenase is an enzyme. Got the point? Succinate is a substrate, fumarate is a product. You have succinate dehydrogenase is an enzyme. The inhibitor is malonate. They'll ask you what is the inhibitor for this reaction. So you should know malonate is an inhibitor. So if inhibitor binds to enzyme, you will not get product. If succinate binds to enzyme, you get fumarate, and succinate and the malonate are similar in shape. Got the point? Are you all there? I is inhibitor. That is para amino benzoic
And please scroll up a bit.
Yeah, these are the examples. If you have any difficulties, please ask me.
Ma'am,
Yeah,
ma'am, what is that word you written after para amino?
benzoic acid.
Okay, ma'am.
Done? Okay, good.
Yes, ma'am.
Okay. So now, non-competitive. Non-competitive. Inhibition.
Excuse me, ma'am.
Yeah.
Ma'am, when inhibitors combine with enzyme, with the product, we also get the enzyme, no?
Yeah, you get enzyme back. Enzyme is unchanged. That's a property of enzyme, which I mentioned you earlier. Enzyme will remain unchanged.
Yeah, so we did not show it with the product.
See, there's enzyme, when it binds with substrate, you get your product. Okay. Enzyme, when it bind with inhibitor, you will not get your product, what you want, what you are applied for. There is no formation
Okay,
of
ma'am.
product because substrate is not for the substrate. See, when we want to prepare a coffee, we add coffee powder, right? If we add tea, do you get coffee in return? No.
Yes, ma'am, got it. Thank you.
You're welcome. Now, non-competitive inhibitor is you have structure of structure structure of S and I are dissimilar. You have enzyme plus you have substrate and you have inhibitor. I mean, let me draw the inhibitor something else. This is inhibitor. Do they look similar? Do they look similar? Substrate and enzyme, substrate and inhibitor are looking similar. Simple question I asked you.
No, ma'am.
Ma'am.
Very good. First understand and then you write. Okay, please understand. Here what happened, your structure is there, that is your enzyme is, this is enzyme, that is substrate and inhibitor. Substrate and inhibitor are dissimilar, but still there is a for ability of an inhibitor to bind. It might not bind to the active site, but it might bind to enzyme. So who is removing now? Who's been removed now? Some devil is removing Ahmad. Who is this devil in the class? See, the reason I schedule by myself, I cannot change the settings is because I use Apple Series, so I cannot do it. I have to inform the office to do that, to do the changes. I cannot do it here. And you people want me to do this extra job, calling Ma'am Fina to reschedule the class and put everybody into attentive and all. Why can't you be quietly sit and listen to the class? I cannot do this from neither from my system, neither from my iPad. I don't have that option. Otherwise, from next time, whenever I have a class with you guys, only with you guys, this problem is happening. No, it's not happening in Pranil in my system. Neither Apple Series is not allowing me to do that. I checked so many times. I have to inform Ma'am Fina for each and every time.
Maybe you have to download Chrome browser.
Chrome browser. Let me check. Wait. Wait, let me check.
File services from Chrome.
Chrome. Okay, I'll go to the Chrome browser and I'll see how it is. I'll do this later. And you people start doing this, is so bad, man. How many times I've been telling you, don't repeat this, don't repeat this. Why you don't understand this simple thing? I know that everybody is not doing it, but you people have to listen. But the one person who is doing it, it's really bad. Okay, let it get downloaded. Okay. So what I was saying, the enzyme will not, the substrate will, and enzyme, substrate and the inhibitor are dissimilar. They are not similar structure, but inhibitor binds to enzyme that cause a conformational change. Since there is a conformational change in the enzyme, substrate cannot bind. When substrate cannot bind, there is no product. Did you understood this? Yes, disfigured. So the active is disfigured. You're getting the right way. Yes.
Ma'am, please scroll up.
Yeah, sure. 2 minutes. Huh? Let me write down this. I'll give you time. Is it okay now? Still more.
Okay, ma'am. Done, ma'am.
Is it done? Okay, good. Any doubts? Now read. Okay, any doubts? Okay, before I allow you to read further, let us understand some important examples. Examples for non-competitive. Cyanide, first example, cyanide poisoning on cytochrome oxidase. See, what they give is they do not ask you the reaction, nothing. Okay, they'll ask you the cyanide poisoning on cytochrome oxidase is a competitive or non-competitive or any other form. So you should know which form is it. Is it clear? My voice is not clear. Sanika, my internet is okay, good enough. Everybody is facing the same problem.
No, ma'am.
Okay. Sanika, you can just re-log in and check because my internet is full. It's on full. Okay, Sanika? Okay, thank you, Shifa. What is proenzyme? That's just one example that you have to know under non-competitive. Proenzyme or zymogens. These are inactive enzyme. Example, pepsinogen. Pepsinogen is active or inactive? Pepsinogen is active or inactive? Very good. It's inactive. What is the active form of pepsinogen, Mangesh? What's an active form of pepsinogen, Mangesh? Pepsin. Very good. Now tell me which is the largest enzyme and smallest enzyme. Who can answer this? No, you do not use Google, but if you know, you tell me. Otherwise, I'll tell you. Largest enzyme is catalase. The smallest enzyme is peroxidase. Next, you have the third one is competitive, non-competitive, and also there is one more called allosteric or feedback inhibitors. Okay, here write down example. First, we'll write down example: glucose plus ATP in the presence of enzyme glucose. It form a
Could you please put
glucose 6 phosphate plus ADP. The enzyme here is hexokinase is an enzyme. This is substrate and this is product. The product will go and stop negative feedback. Okay, here product is acting like inhibitor when it's excess. Please
The
write
screen
down.
is not visible.
Screen is not visible to anyone.
Ma'am, please scroll up.
Please re-log in. I think student can see the screen. Can you see the pro enzyme? Please re-log in. Write down. Okay. Are you done? Next, write down one more example for non-competitive. Okay, this is also important. I missed this non-competitive example under non-competitive. Okay, acetylcholine is a neurotransmitter. Acetylcholine is a neurotransmitter. Is a substrate. Acetylcholine esterase is an enzyme. You have acetylcholine, that is active acetylcholine. Active acetylcholine. And here you have an inhibitor that is called DFP or nerve gas. First write down, I'll explain you how it works. DFP full form is di-isopropyl fluorophosphate. See, listen to me. Neurotransmitter, during the breathing, neurotransmitter are released, and this neurotransmitters goes and contract the diaphragm, and then the acetylcholine will convert or activate the acetylcholine to release the diaphragm, so that the diaphragm is relaxed and this breathing continues, like contract relaxing continue. If nerve gas or DFP, which is a inhibitor, if they come and bind to acetylcholine, they cannot convert the diaphragm, which is contract, to relax. Here the diaphragm is contract, and here under the presence of enzyme acetylcholine esterase, it get relaxed. If DFP comes and bind to acetylcholine, then the diaphragm cannot relax and the person dies. Is it clear to all? Now, I want each and everyone to read about competitive, non-competitive feedback inhibitions. Quickly.
Excuse me, ma'am.
Is it done? Any doubts to anyone? Can we move next? Can we move next?
Excuse me, ma'am.
Yes, tell me.
Ma'am, could you please explain feedback inhibitor part again?
Yeah. See, whenever in a body, see, the substrate is converting into product, okay, by the help of enzyme. Whenever there's excess of product which is increasing, keep on increasing, this product itself go and stop the enzyme. That is called feedback inhibitor. See, for example, you're having a breakfast and your mama is making some rotis. So you keep on eating, right? And once you feel satisfied, you yourself tell mama, I'm done. I don't need more rotis. It's kind of thing. Did you got the point? So the product itself send a negative signal to the enzyme to stop converting substrate to product because there's too much product already in the body. Got the point? Whose question was that?
Yes, ma'am.
Yeah.
Yes, ma'am, got it. Thank you.
You're welcome. Any other doubts to anyone? Can we move next? Are you all there? Am I audible?
Yes, ma'am.
What has high probability? I didn't get the question. So now we are studying the classification of enzyme. Important again. So write down, make 4 columns. Serial number, EC number, name, sorry, 5 columns, name, function, and example. EC number, you can write down the full form of EC is enzyme commission. The first one is Roman number 1. Name is oxidoreductase enzyme. They help in oxidation and reduction reaction. Help function is help in oxidation and reduction of reaction. For example, you can give any example which you come across. Catalase do this, oxidase does this, peroxidase does this. So these are the examples that bring about oxidoreductase. Okay, they are come under the category of oxidoreductase. Second is transferase. You have transfer of functional groups. Transfer of functional groups.
Ma'am,
Yeah.
is that RECU?
RECU. Reductase. REDCU. DUC. Which one? Reduction.
No, ma'am, the oxidoreductase.
Reductase. CUTASE. Reductase.
Okay,
Clear?
ma'am, thank
Amino transferase. Kinase comes under this category, such as hexokinase. They come under this category of transferase. Next, third one, hydrolase. Cleavage of bond with addition of water molecule. Cleavage of bond with addition of H2O. Breaks large molecule to small molecule. means it break large molecule or a bond. They break bond or molecule by with additional of water molecule, such as protease. Peptide bonds are broken by protease, amylase, all this. Protease, lipase, the lipase break ester bond, amylase, they break peptide bond. So all this comes under the category of hydrolase. They break the bond with addition of water molecule.
Ma'am,
Hmm.
ma'am, the second one, that example, is it one example or it is two?
It is this one, this is one, and this is two.
Okay, ma'am.
Aminotransferase and kinase.
Okay, ma'am.
The fourth one is lysis. Break bond without adding water. Without adding water. Example, aldolase, isomerase. Interconversion of isomers. Exoisomers or exoisomerase, that is change glucose to fructose. One example. Next. Sixth one, ligase. Use ATP join small molecules to form bigger molecule. Example, DNA ligase. I have a good news for you. Are you do it and then are you done?
Yes, ma'am.
Okay. Now go through it. Go through it. Go through the table. Everybody go through the table.
Ma'am, what were the NEET results this year?
We cannot disclose it, but yes, it will be on 16th of this month. So we'll all let you know.
Ma'am, it is on 16, no?
16th, yeah, hoping to be on 16th. So if it's on 16th, we'll be having a very good result. This will be disclosed on 16th. We cannot tell you now. Okay. Yes, 16,
ma'am.
16. Please go through this quickly. Done? Any doubts to anyone? Okay. So the good news is that we have finished this chapter, the biomolecule, and we are completing one more chapter today. The big chapter again, where you will study about carbohydrates, proteins, lipids, nucleic acids, different forms, and finally enzymes. So with this, we are completing a biomolecule chapter. How did you find this chapter? Easy, moderate, or difficult?
Moderate.
Moderate. Okay. See, you can make it easy. You can make it easy if you sit and study this together. I mean, together means at one stretch you read carbohydrate, at one stretch you read protein, at one stretch you read lipids, then it will be easy. So I'll give you 5 minutes, quickly revise the content. If you have difficulties, please ask me quickly about the enzyme. So next test. Oh, okay. So next test will be on animal kingdom, right? Conjugate enzyme. Okay. And what else? What are the doubts? Okay, let me draw it here. See, for example, if this is your enzyme, if this is your protein, okay, if this is your protein, to this protein, you're attaching some bond with the help of some bond, you're attaching some enzyme. Now, this is called conjugate. This is an antibody, for example. This antibody is for, whose question was this? Let me check this question.
Ma'am, me Arya.
Arya, listen Arya. So you have an antibody which is protein. To that antibody you are adding an enzyme. Clear? Now, this antibody is from the patient. Not from the patient, sorry. This antibody is for HIV. This antibody is for HIV. To that you are adding an conjugate enzyme. And this enzyme is what type? Conjugate, because it is attaching to protein, that is antibody. Now what you do? You add the blood sample of HIV patient. When you add blood sample, if that antigen is showing positive, then this enzyme will convert the substrate into a different color, that is called product, if that person is positive to HIV. If that person is negative to HIV, it will not give you a colorful product, it will remain the same. This is how they use it in the medical field. This we'll study further in 12th standard, you do not have to know, but since you wanted to know how the conjugation takes place, if this is a protein attached to enzyme is called conjugate. Clear? Okay, next. Arya, is it clear? Next, ma'am's feedback inhibition. Sanika, not Sanika, Sania, right? Sania. Sania, listen. The S is substrate and P is product. E is enzyme. Substrate convert into product in presence of enzyme. I hope this part is clear. Now listen. What happened? Your product will go and stop the enzyme, telling the enzyme not to convert substrate into the product because you're already having too much of product in your body. That is called feedback. You're giving your feedback in a, like, how do you give feedback? Same way. As I recently quote that example, like you're sitting for your breakfast and your mom is making rotis for you. She makes and get you rotis. Once your stomach is full, you tell your mama, stop making rotis, I am done for the breakfast, right? You're giving your feedback to your mother saying that do not make rotis because I am full now. Did you got the point, Sania? That is called feedback inhibition.
Yes, ma'am, understood.
Okay. Any other doubts? Structure of cholesterol. I don't know whether I have in this slide. Let me check. Structure of cholesterol. Yeah, you can take down the structure of cholesterol.
Ma'am, we should write this.
No, don't write this. What happened? People understand faster when food is used in example. Okay, fine. Is it true, Sania, what Amod is telling? Okay. So I hope no doubt. So which lesson do we shift now? Which lesson do we shift now? Can we, ma'am, will it be possible to share PDF? No, I cannot. Sanjana, I cannot. I cannot share the slides. Yes, circulation. That's the reason.
Excuse me, ma'am.
That's it. I would have given Sanjana. I would have given you the PDF long PPTs long time, but since not allowed, that's the reason I told Sanika to share the notes, which the other student missed. Yeah. Okay.
Excuse me, ma'am.
Yeah. Tell me.
Ma'am, could you please show the last slide which you showed just now?
Cholesterol slide.
Yes, ma'am.
Okay, blood. I think most of the things you already know about the blood, fluids, and circulation. The next issue, the next chapter, very interesting chapter. So we are tired of reading biomolecules, animal kingdom. Now let's move to the some human physiology part, right? Let's move to the human physiology part. Previous slide, which one? Which one? Which is the previous slide you are saying, Sanika?
No, ma'am. I saw it just now. The screen is lagging. That's why.
Okay, fine. Okay, are you done? Haman, I'll tell you when to join the Darwin class whenever there is enzyme
ma'am.
part. I'll text you. You can text your friends. Is it clear, Prabhu Desai?
Yes, ma'am.
That's what I said. I'll teach the part that is missed. That's the only small part what you have missed. So I'll call you. You can come and some forwarding batch. Why forwarding batch? Okay, you also missed. Okay, the people who are missed that part from enzyme, you all can join. I'll text one of you and then you can talk to each other and can join. Okay, I'll let you know when it is. Is it clear? Okay. Next. Yeah. How much time do we have? Okay, let's brief out about the blood. Tell me, what do you know about the blood? What type of a tissue is it? Let us just brief out about the blood. What type of tissue is it? Connective tissue. Rishikesh say connective tissue. Fluid connective tissue. I need a proper answer. The proper answer, one answer is given by Sakshi and Ash. That is fluid connective tissue. Under fluid connective tissue, what is the other name for the fluid connective tissue? Who can answer this? Who can answer this? Under fluid, what's the other? Very good. Very good, Mangesh. It is vascular connective tissue. Do you remember? So, what are the things that come under a vascular connective tissue? Very good, Sanika. What are the tissues that comes under vascular connective tissue? Lisa, tell me. What are the tissues? What are the things that comes? Two types of vascular connective tissue. Who can answer this? I don't want anybody to Google there. I want your answer promptly because I trust you. If you know, you tell me. Very good, Rishikesh. It is blood and Sakshi. Very good. It is lymph and blood. Do you all agree? Very nice. Among blood and lymph, which is the colored, which is the colored solution? Blood. Why is the blood colored? Why is the blood colored? Because of hemoglobin. Very nice. Next. What is a special feature of vascular connective tissue? Who can answer? What is a special or unique feature of vascular connective tissue? Let me see who will answer this. What? Because of hemoglobin. Yeah, because of hemoglobin. No. The question here. Okay, that was a previous. Okay. Very, very good, Sakshi. I got your answer. Fibers are absent. Very good. So you will not find fibers in this vascular. No fibers. Very good. Now, which are the cells that are present in lymph? Which are the cells that are present in lymph? Who can answer this? Okay, let me ask you like this. What are the formed elements present in blood? Amod, I have given the answer to the previous question. I'll come back to that, Amod. The question here is, what are the formed elements in blood? I want you together, Sakshi. Oh, Sakshi. Okay, I think that is the answer for the previous. Very good. Now the question is, what are the formed elements? Formed elements of blood. Formed elements. Mangesh, I'm talking about elements, not plasma. Plasma is a fluid. Okay. Formed elements is RBC, WBC, Sanika, very good. Erythrocytes, leukocytes, thrombocytes, Lisa, very good. So those are among erythrocytes, leukocytes, and thrombocytes, which take place or take part in clotting. Among all these three, which take part in clotting? Platelets, very good. Thrombocytes, very good. Very nice. Next. How much liters of blood is present in the healthy human body? How many liters? 5 liters. Very nice. You people have become very smart, huh? That's nice. Very good. Next question is, what's a pH of blood? What's a pH of blood? Yeah, even a bile is right. It can be from 5 to 6 liter. Okay. It can be 5 to 6 liter also. pH is 7.4. Very nice. 7.4. 7.4. Very nice. Okay. So now tell me, in lymphocytes, in lymph, what type of cells are present? WBC, platelets, very good. They have WBC, they have platelets, they have TB cells, but they do not have RBCs and the platelets. Okay, next. They do not have RBCs. Where this, which are the two types of granulocytes? No, agranulocyte cells. Which are the two types of agranulocytes? Which are the two types of Granulocytes, lymphocytes and granulocytes, monocytes. Very good. Lymphocytes and monocytes. Which are the three types of granulocytes? Okay, so Abail, what do you want me to do? You want me to send a power? Abail. So it's the lymphocytes and neutrophils, eosinophils, and basophils. Yes. Very good. Any doubts? I'm really happy that you people answered so well. This was taught to you in the first chapter, right? If I remember, this everything was taught to you in the first chapter and you all answered so well. That's really nice. Now tell me, yes, the first chapter. So next class will be continuing with this, but before this, I want you all to learn about body fluids, the blood part and the lymph part from the vascular system. Okay, in the next class. Have we done the blood grouping to you all? Have I done the blood grouping? Have I done the blood grouping? No. Okay. So you learn about WBC, RBC and platelets and about the lymph. Just revise yourself. Already you know many things about the content. Then we'll start about the blood grouping in the next class. Clear, my dear students? Okay? Yeah. So enjoy your meal. See you all in the next class. Until then, keep learning. Bye-bye, everyone.
Thank you, ma'am.
You're welcome. Bye-bye.